Good morning, students. In this experiment, we are going to learn how can we separate plant pigments through paper chromatography. Okay. So, in the smaller classes, you studied which are the different types of plant pigments: chlorophyll, xanthophyll, carotene. All these are known as different types of plant pigments. So here we are going to separate chlorophyll A, chlorophyll B, xanthophyll and also carotene. Okay, four pigments we are going to separate through paper chromatography. How can we do that? So these are the material required for that. We need one motor and vessel, and we need beaker, and you can also see this is the capillary tube. By using this we can take this extract, and we need some Batman filter paper. This is a filter paper, this is known as Batman filter paper, and we need two solutions. One is acetone, then second one is, this is petroleum ether. Okay, the solution is petroleum ether. We need one measuring cylinder, and also we can make one chromatography setup, chromatography tube by using this test tube. I'll show you how can we make it. We can close this by using one cork. It's a split cork. You can see the split cork, and one paper. I'll tell you how you're supposed to make this paper. Paper in one end it is tied with a thread. Clear? So by using this, so we will close like this. So this is the chromatographic tube. So first what you are supposed to do, you have to take some leaf. You have to take spinach leaf or any leaf, green color leaf you can take. So I have taken this leaf I have taken from this garden. So you can just make it pieces and put into this mortar and pestle. By using this, you have to crush it very well. So while crushing time, so I hope you can see this, you have to crush it well. You can add little acetone into this. Okay, so add little acetone solution into this, and you have to crush it well. Clear? Then after crushing, what you will get that juice or extract you will get. That extract slowly you have to pour into, by using the funnel you can pour into another beaker. From this beaker, if I need to take this extract, I have to use this capillary tube. Okay, so this is ready here. The next we have to make one solution. Two solution you have to mix it. So which are the solutions? So one is petroleum ether. So there is one ratio, petroleum ether and acetone. The ratio is 9:1. So 9 ml petroleum ether and 1 ml acetone, both you have to mix it. Okay. Then after mixing, you have to take this solution in this. Already I have taken the solution in this test tube. This is actually chromatography tube. So this chromatography tube means I have taken one test tube, and you can close with this. I told you this fork. It's a split cork. This is a cork. This is a split cork. By using this, you can close this test tube. In between this cork, what you are supposed to do, you have to put this filter paper. Filter paper, how I cut this, you see this, it's like a rectangle shape. In one end, we made like a wedge shape, little narrow end. Other end, I tied with this thread. Clear? Then, what is supposed to do? Now the solution is here. Which is a solution? Acetone and petroleum ether. Both I mixed, I taken in this. The ratio is 9:1. 9 ml petroleum ether and 1 ml acetone. Both you have to mix, and it's ready with this chromatography tube. Okay. Then this paper, I told you this is the Batman filter paper. This filter paper you have to cut it. So I already cut. How you are supposed to cut it? I told you can make it a little long rectangle shape, and one end you can just make like this wedge shape. Then just from down 2 cm just above, you can draw a line by using pencil. Use one scale and pencil, just draw a line. Okay, this line is called loading spot. What it is called? Loading spot. Here you are going to put this juice. Okay, or extract. So what is supposed to do by using this filter paper? Using this capillary tube, I am going to take this chop. I am going to put one chop in this line. So I already showed this is the line A2 in this paper. So I will put just one chop in this. Okay, you have to leave like this, then it will dry. After 5 minutes it will dry. Again you have to take little more extract. Again you have to put into this the same place, that line I drew, that is called loading point. Again second chop. Same like you can do 3 or 4 times, 3 to 4 times you have to put this chop, and you have to leave like this. So after some time what will happen, it will dry. Okay, so one time you put one drop, then 5 minutes you leave, it will dry. Again second drop, then third and fourth, then it's dried nicely. Then this paper I am going to put into this. This is a chromatography tube is ready. This is a split cork between. So one end I tied with the thread. I am keeping this thread like this. I will hold tightly. This paper I am going to put inside this. Little more like that. So we can put like this. Yes. It should just touch. Okay. It will just touch the solution. You can put and adjust it. Yes. Okay. So this chromatography paper, you can see that it touches the solution. So what is the solution? It is a mixture of acetone and petroleum ether. Then you have to leave like this. After something you can see what you can see. The solution, it comes up and it will reach till here. So when it reaches, you will be getting here. So already we put 3-4 drops of pigment here. That pigment, it also will spread along with the solvent. You will be getting different color bands. You will get first green color, then bluish green color, then yellow color, then orange color, like that you will be getting different color bands. So which is the bluish color? That is chlorophyll A. Chlorophyll B. Chlorophyll B, the next one is chlorophyll A, that is first green color will come. Green is chlorophyll B, the next bluish green, chlorophyll A, then xanthophyll, yellow color, then carotene, orange color. Like that you will be getting different color band. Then you have to find out, so you have to keep like this for 10 to 15 minutes. Then after some time slowly you will be getting the color bands. You are going to find out the RF value of this pigments. How can you find out the RF value? RF value means the distance traveled by the pigment divided by distance traveled by the solvent. So suppose we are taking the first pigment, maybe it will reach till here. The distance traveled by the pigment, it may be 2 cm. We have to measure it 2.1 or 2 divided by distance traveled by the solvent it will reach till here. So that may be 8 cm you take. So 2.1 divided by 8. You will be getting the answer. What is the RF value of chlorophyll B? Same like next one is chlorophyll A, second pigment. Next color band, from this line till the second pigment you see what is the distance you have to measure by using the scale. So you may get sometime 3.5. So that value divided by the distance traveled by the solvent. So that is the formula for RF value. Okay. So by using this you can find out the RF value of each pigment. Distance moved by pigment divided by distance moved by the solvent. You have to find out. So in your book, this book, it is given chlorophyll B, green color, 0.42 RF value. Chlorophyll A, bluish green, 0.6. Xanthophyll yellow, 0.72. Carotene yellow orange, 0.95. Like that you have to find out the RF value of each pigment. Clear? So this is a way we have to separate plant pigments, different plant pigments through paper chromatography. Thank you, students.